Good morning. Welcome back to Biology 102. Today we're looking at genetics and Gregor Mendel. We will be looking at Mendel's 2 laws of inheritance and a couple of devices that allows us to track inheritance. Remember, inheritance is the passing of traits from parents to offspring. Over here, I have a list of terms that will be in this lecture. They are also on your review sheet. I highly recommend that you look over these terms. Genetics is a bit like a foreign language, so you need to practice that vocabulary. All right, Gregor Mendel is known as the father of modern genetics. He was a monk or a friar that lived in the 1800s, and he studied pea plants. Now, he asked an age-old question. Even in the 1800s, I'm sure people were asking this question. Why do offspring look like their parents? Right? This is an old question, but Gregor Mendel was the first scientist to really make some breakthroughs on how traits were being passed from parents to offspring. Now, why was Gregor Mendel so successful? I think he had lots of things going on in his favor. He chose purebred pea plants. He knew how to do this because he was from an agricultural background. He chose plants that were quick and easy to grow. He chose traits that he could see with his naked eye. He meticulously crossed them by hand and controlled his experiments. And he knew a little bit of math, which comes in handy for genetics. Gregor Mendel left us with 2 laws of inheritance. The first law is known as the law of segregation. The second law is known as the law of independent assortment. Before we get to these laws, I want to talk through some terms with you. So the first terms, traits in genetics, are any heritable feature that passes from a parent to an offspring. Homologous chromosomes, like in blue over here, are 2 chromosomes that carry the same sequence or set of genes. However, those genes can come in different versions. Those are known as alleles. So up here on my chromosome, I can have the gene for flower color in the same position on my homologous chromosomes, but one of these chromosomes carries an allele for purple flower color and one for white flower color. These 2 different flower colors or forms of the flower color gene are known as alleles. Finally, we often will abbreviate the parental generation with the letter P and filial or offspring generation with F1, F2, etc. All right, Mendel lucked out. He chose pea plants as his study population. And for Mendel, a lot of the traits in peas were quite simple. They would carry alleles, different versions of genes, but they would come in 2 different forms. Either dominant alleles, which always show if you have one dominant present. We often designate this or give it as an uppercase letter. And recessive alleles. Recessive alleles only show if there's no dominant alleles present, so you need 2 copies of this, and we often show them as the lowercase letters in our sort of diagrams. Now, I show freckles over here. Freckles is a dominant trait, so if you have 1 or 2 copies, you will show freckling. And the way freckling works is if you have melanin sort of evenly placed, you will just tan evenly across your skin, but some people have it in patches so that you get these sort of freckling spots. It is a dominant. There are some things that can sort of influence it, but it is a dominant allele, so if you inherit 1 copy, you will show freckling. And to be recessive without freckling and even tanning, you need 2 copies of that recessive in order to not show freckles. The second thing that Mendel found was that he found that for each trait, an organism inherits 2 alleles, 1 from each parent. And we know this because of homologous chromosomes. You know that 23 of your homologous chromosomes come from your dad and 23 of your homologous chromosomes come from your mom. But Mendel also named the states at which people sort of carried their alleles. So if you had 2 alleles that were the same, Mendel called you homozygous. Homozygous dominant could be like purple flower color. If you inherited a purple on this chromosome and a purple on this one, you would be homozygous purple for flower color. Homozygous recessive might be 2 lowercase letters. So how about white flower color and white flower color? If you inherited 2 whites, which are recessive to purple flower color, then we would say that you are homozygous recessive. Heterozygous is the state where you inherit 2 different alleles. So up here on our chromosomes, it's like capital B and lowercase b. This is where you might inherit a purple flower color from your mom and a white flower color from your dad. This brings us to Mendel's first law, the law of segregation. The law of segregation states that two alleles for the same trait segregate or separate during gamete formation, and they end up in different sperm and eggs. I want you to underline that word same trait in your notes because in this particular law, we're talking about alleles for one trait, so purple flower color or white flower color. If you were a heterozygous person who inherited, or pea plant, that inherited purple flower color from mom and white flower color from dad, so you are heterozygous, then this means that half of your sperm and eggs would carry purple color and half would carry white color. Now, Mendel figured this out not only for flower color, but a lot of different traits. And you don't need to memorize these, but he did it for many different traits inside of his pea plants. Now, his second law is known as the law of independent assortment. And this law says that pairs of alleles assort independent of other alleles for different traits as well in gamete formation. This means that if you had something like hair color, let's pretend some of these genes over here are hair color and some are eye color, that hair color and eye color would assort independent of each other when we form sperm and eggs. We know this to be true because of crossing over. We know that when you have two traits on your homologous chromosomes, they can recombine during crossing over for these different traits. And so traits are not stuck together, right, in these like sets. And Mendel noticed this as well. Mendel noticed this because he did dihybrid crosses, where he would cross two and track two traits at once, or two allele forms at once. So over here we have a round shaped pea with a yellow color, and then a wrinkled pea with green color. And he would track these two traits at once, and he noticed that you didn't get a simple pattern where yellow and round got stuck together, but rather each of those traits, roundness and then the color, yellow or green, would be tracked independent, and you would get this like combination of traits for your dihybrid crosses. So this led Mendel to say that alleles for different traits also segregate independent of one another in gamete formation. So working beyond Mendel, there are a few devices that help us track inheritance. One of those is called a Punnett square. These are devices that allow us to predict genotype and phenotype frequencies in offspring given the parents. So kind of doing what Mendel did before. In these Punnett squares, we place the parents on the outside of the box, and the offspring potentials are in the inside boxes. Now, genotypes are these two-letter combinations, right? So the allele combinations for an offspring. And phenotype is a description in words of the appearance of the trait, like purple flower color or white flower color. We will be doing some examples of these in class in our tutorials, so join me for those. The other device I want to mention is a pedigree, and pedigrees are like family trees that help us map out a trait and how it's being passed in a family. Pedigrees also have rules, just like Punnett squares. So in a pedigree, we line up our generations. So you'll see the generations. Typically, if you are an XY 23rd set, you will get a square. If you're an XX, you will get a circle. Any trait that we are tracking gets colored in. If you don't have the trait, it gets open or left open. And then two different parents, any offspring they produce comes down to this generation, and we typically list our offspring from left to right in order of birth. These devices, again, we'll be practicing in our tutorials. You can take a look at the notes, but also see me there. Thanks again for watching me for Mendel and inheritance, and I will see you again next time.